Here we go, part 4 of chapter 6. So we left off talking about vesicles, and I just wanted to help you define what a vesicle is. So basically, it's a sac. It's enclosed by a thin membrane, and most vesicles have a specialized function assigned to them. They can be separated from the cytoplasm by either one phospholipid bilayer, or they may have two or more even. So it really depends on the function of the vesicle. Some of those functions are storage. There are transport vesicles. There are vesicles that do digestion of waste products. That membrane that encloses the vesicle is very similar to the outer phospholipid bilayer of the cell. So the membrane of a vesicle can then fuse with the plasma membrane, allowing the vesicle to release its product to the exterior of the cell, so for secretion. Since the vesicles are separated from the cytoplasm, sometimes they can hold different conditions inside their vesicles. So, for example, again, I talked about this a few minutes ago, they can have a different pH, for example, or they can contain enzymes that would be doing digestion of the cell organelles if they were free to roam in the cytoplasm. So the different types of vesicles, again, can be lysosomes, transport vesicles, secretory vesicles, meaning they secrete products to the outside, or vacuoles. So I want to look at the vacuole in particular. So vacuoles are found in both plant and animal cells, and it's a type of vesicle with a specialized function depending on where it is. So if you have a vacuole in an animal cell, it's usually very small. Common vacuoles that you will find in an animal cell would be phagocytic vacuoles. So that is a vacuole that engulfs something in order to digest it. And so it engulfs it and closes it within it. It eats it, essentially. Food vacuoles, so food storage vacuoles you'll see in animal cells. Some animal cells have what are called contractile vacuoles. So there are single-celled organisms that use a contractile vacuole in order to move around. So they don't have cilia or flagella to make them move. Instead, they can pump water in and out of their vacuole, contracting it in order to push themselves along. Where you really see a large vacuole is in a plant cell, and they serve a lot of functions in a plant cell. So it's a big part of what gives structure to a plant cell, especially a parenchyma cell, which is involved in photosynthesis and is found in the leaves. So inside a vacuole, usually vacuoles are filled with liquid, so they're usually water-filled. In a vacuole in a plant, that fluid is called cell sap. And in a plant, the large vacuole has mineral salts, sugars, organic acids, oxygen, carbon, carbon dioxide, pigments, and waste. So it is really the storage center for the plant cell, for all the different nutrients and waste that the cell is using. Water enters a vacuole by osmosis because the vacuole has a semi-permeable membrane called a tonoplast. When water enters the vacuole, it actually puts pressure on the tonoplast. So the tonoplast is not like the cell membrane. It's not as fluid. It actually has more rigidity to it. So it's almost closer to a cell wall. And so when water is pumped into or through osmosis goes into the vacuole, it actually puts pressure on that vacuole. And then the vacuole expands, and as it does that, it actually puts pressure on the cytoplasm and pushes the cytoplasm of a plant cell up against its cell wall. And so by doing that, it's actually helping the cell either get a signal to grow, or it's helping the plant actually have the ability to stand upright, for example, in order to have access to sunlight for photosynthesis. So it's maintaining a normal water balance within the cell, and particularly the plant cell. So that vacuole has a real key role in the plant cell. Also inside a vacuole, kind of a side note, is where you find the pigments that are responsible for the color of flowers or fruits or the buds or the leaves. So those pigments of color that we see are actually found in the vacuole. Leaves actually get their color also from the chlorophyll in the chloroplasts, so a different organelle in the cell. One last thing is that a plant vacuole also is where you'll find the food reserves, like sugars that the plant cell needs, are going to be stored in that. And also waste products are stored within the big plant vacuole. Another type of vesicle is a lysosome that has a very specific job. So a lysosome is a phagocytic organelle, or actually not really an organelle even, a phagocytic vesicle. It actually will engulf something and digest it. So lysosomes are filled with enzymes. Some of them will be filled with very low pH, so they're very acidic. But most lysosomes use enzymes to recycle old matter, like old organelles or macromolecules that don't need to be used. Lysosomes will basically break them apart using enzymes. Another type of phagocytic vesicle is a peroxisome. So these are very specialized as well. They produce hydrogen peroxide and convert it to water. So peroxisomes are where you would find the enzyme catalase. So catalase needs to be within the peroxisome in order to break down the hydrogen peroxide. And so peroxisomes are very important. Okay, so the next topic I want to cover is the endosymbiont theory. And so there is a theory out there that there are evolutionary connections between mitochondria and chloroplasts and bacteria. So the thought is that mitochondria and chloroplasts, those organelles, actually at one time were simple bacteria that lived outside of a cell. They were autonomous bacteria organisms. And what happened is they were enveloped by another bacteria. You see that picture here. And they became part of the bacteria that ate them, essentially. But they were able to still function, and actually the bacteria that ate them benefited from having them in there. So there was basically a symbiotic relationship that occurred where we get this endosymbiotic theory. So endo meaning brought within, symbiotic is symbiont. So that's where we get the word here on this theory. And then over time, the bacteria was passed down, the internal bacteria that became the mitochondria or the chloroplasts, was passed down from generation to generation of the succeeding generations because it was a favorable adaptation. Part of the theory behind this, you can see here, is there are a lot of similarities between bacteria and mitochondria and chloroplasts. So one of those is that mitochondria and chloroplasts have their own DNA. and their DNA is not in the nucleus, but it is in a nucleoid region. So both mitochondria and chloroplasts have DNA. This next picture shows you some more similarities. They also have a smooth outer membrane and an inner membrane folded into cristae, which is very similar to the middle here. We have a picture of a bacteria. Very similar structure. You can see this idea of cristae and in the bacteria and cristae in the mitochondria. The other thing is that they have free ribosomes in the mitochondrial matrix, and bacteria have free ribosomes as well. This is a picture showing how mitochondrial DNA is found. What this picture is showing, just so you don't get confused, is an actual cell. And so that's why it's showing nuclear DNA, because this is a eukaryotic cell that has a nucleus with DNA right there. And what they're trying to show you here is the relative size of a cell's DNA compared to, these are actually mitochondria here, that are showing their DNA, a much smaller scale of DNA, which again is very similar to a bacteria's DNA. Okay, and this picture is just showing how a chloroplast in its structure is. And when we look at photosynthesis, we're going to look a lot more closely at both this and then cellular respiration. We'll be looking at the mitochondria. So we'll go into a lot of detail at that point on the structure of these two organelles. So the next thing I wanted to talk about, actually, before I do, I'm going to tell you a joke because I feel like listening to me lecture must get very difficult late at night. So I'm going to add jokes every once in a while. So on this joke, did you hear about the famous microbiologist who traveled in 30 different countries and learned to speak 6 languages? Well, he was a man of many cultures. Get it? Cultures? Like microbiologists make cultures of bacteria. Ha ha ha. Okay, so now the next concept that we're going to look at is cytoskeleton. And what that is a network of fibers organizing the structure and activities in the cell. They used to think it was just making up the structure of the cell. So skeleton, you think of structure. But what they've learned is that the cytoskeleton is actually involved in transport within the cell. So it's made up of a network of fibers that go all throughout the cytoplasm. It anchors a lot of the organelles, so it almost acts as like a web to hold the organelles in the correct place. There are 3 types of molecular structures that make up the cytoskeleton. So just be aware those 3 things are microtubules, microfilaments, and intermediate filaments. So those are the 3 main parts of the cytoskeleton. And the main job of cytoskeleton is support and also motility, so the movement of a cell and the movement within the cell. So for example, hopefully we're going to have a time to look at the inner life of a cell, which is a video that was just incredible. If you have never seen it, you must see it. It is animation that shows, pulled together all of the current knowledge about what the inside of a cell looks like and how it works. And it just totally revolutionized how we look at the internal structure of a cell. And one of the things that's really neat in it is the depiction of motor proteins, which are proteins that actually walk along the cytoskeleton carrying vesicles for example, from the endoplasmic reticulum.